The COVID-19 pandemic has highlighted the importance of vaccines in protecting against infections or in reducing the severity of symptoms among vaccinated individuals. Vaccines typically contain a harmless form of a virus or bacteria known as the antigen. Upon vaccination, activated T cells provide help to B cells, which go on to differentiate into antibody-secreting plasma cells. These antibodies are key to conferring protection against an infection due to their ability to neutralize virus. Vaccines are also important in generating memory cells, which can generate a faster and more potent response during an actual infection. Vaccine-induced antibodies can arise from two main pathways. Antigen-activated B cells can differentiate into short-lived plasma cells early in the immune response via the extrafollicular pathway. Alternatively, B cells can enter the B cell follicle where they undergo class switch recombination before entering the germinal center response. The germinal center is a specialized microenvironment which is composed of two compartments: the dark zone, where B cells undergo clonal expansion and somatic hypermutation of their immunoglobulin variable genes, and the light zone, where B cells receive antigen from the FDC networks and undergo a process of selection. B cells that receive positive selection signals from T follicular helper cells will either re-enter the dark zone for further proliferation and somatic hypermutation, or exit the GC and differentiate into long-lived antibody-secreting plasma cells or memory B cells. Unfortunately, during aging, vaccine efficacy is reduced, which means that the vulnerable members of our population are also the least protected. This age-related decline is characterized by a quantitative decline in vaccine-induced antibody titers and a qualitative decline in terms of reduced antibody function and reduced diversity of antibody genes. In order to find ways to improve vaccine responses in older people, the first step would be to understand key contributing factors underpinning this phenomenon. Multiple mechanisms underlying the age-related decline in humoral response have been characterized, including defects in T cell priming by aged dendritic cells and dysregulation in stroma function. However, whether B cells intrinsically are defective in their function with age remain unclear. In this paper, we aim to investigate if there are cell-intrinsic defects in B cells' response to stimulation or vaccination with age, using a combination of human in vitro studies and mouse in vivo studies. To understand if B cells from older people are as functional as those from younger people, an in vitro assay was set up where naive and memory B cells sorted from the peripheral blood of younger and older people were cultured with T cell signals. After 6 days of culture, we found that B cells from older people were not defective in their proliferative capacity, plasma cell differentiation capacity, antibody secretion, and their ability to upregulate co-stimulatory molecules. This suggests that there are no intrinsic defects in B cells from older people in responding to stimulation. We then went on to investigate if there are cell-intrinsic defects in B cells' function in vivo, using the switch-tail adoptive transfer system. Hennig lysozyme or HEL-specific B cells from either young or aged switch-tail mouse were transferred into young congenitally distinct wild-type recipient mice. These switch-tail B cells contain the high-affinity B cell receptor against Hennig lysozyme. The recipient mice were then immunized with HEL conjugated to sheet red blood cells to elicit a T-dependent response. Similar to what we observed in the human in vitro studies, B cells from the aged donor mouse had no proliferative defect at day 3 post-immunization, compared to those from the young donor mouse. At day 6 post-immunization, B cells from the aged donor mouse preferentially entered the extrafollicular response at the expense of entering the germinal center, compared to B cells from the young donor mouse. Nevertheless, by day 10 post-immunization, when the GC response is more mature, we observed no defects in the ability of B cells from aged mice in mounting a GC response. This suggests that while age-related intrinsic B cell changes delay the GC response, they are unlikely to be responsible for the in-pet antibody-secreting response or smaller peak GC response in aging. To check if B-cell extrinsic factors contribute to smaller GC response during aging, we conducted another transfer experiment where HEL-specific B cells from young donor switch-tail mice are transferred into young or aged wild-type recipient mice. 6 days post-transfer and immunization, we observed a significant reduction in the recovery of young donor B cells and fewer donor-derived GC B cells in the aged recipient mice compared to when cells were transferred into the younger adult mice. In conclusion, in our paper, we show that B cells from older people are not defective in activation, proliferation, and plasma cell differentiation. In addition, B cells from aged mice showed a delay in GC B cell formation early in the immune response, but had no defects in mounting a peak GC response. This implicates B cell extrinsic rather than B cell intrinsic factors as key contributors to the age-related decline in humoral response during vaccination.